Morning everyone, hope everyone's well. We're going to be covering Biology 3 today. Morning Tanishka. So we're going to be looking at infection and response. Probably quite an apt topic with this. So again, if you've got any questions we're going through, put them in the comments box, or I can jump online with a Google chat after this so we can have a bit of a meet. Or if you think of something later on, then just email me. But we're going to be having a look at microorganisms, the pathogens. We're going to be bringing in a certain amount of keywords. I'm going to be having a look at some of our natural defences, some of the different diseases that can be caused. We're going to be having a look at how our body is designed to try and resist becoming infected, and then what happens if it does. So we'll see how much we get through today. Obviously we're going to carry through on Wednesday and also onto Friday as well. So we'll go through from those points of view. So if you do have questions afterwards, I'm more than happy to pick them up during later videos. Now, I suppose as we're sort of looking through this, to start with, the most important thing we're looking at is the difference between communicable and non-communicable diseases. Today, we're only going to be looking at this idea of communicable diseases. So, and let me just put that down here. Ooh, there we go. So, by communicable diseases, we are talking about diseases that can be passed from one person to another. In other words, they're contagious. And I always spell contagious wrong, but let's just put it down anyway. No, I think I've got it right. So we've got contagious diseases. Now, we're not talking about diseases that are caused by lifestyle choices or that are just unlucky. So we're not talking about cancer. We're not talking about diabetes. We're not talking about obesity. We're talking about diseases that can be physically passed from one person to another. So they are generally caused by some form of microorganism. So we can put microorganism down here as well. Okay. We're going to be looking at some of the different types of these, but we need to make sure that we understand that we're looking at anything that is from when start again. We're looking at different diseases that are passed on when a microorganism is passed from a person to a person, from an object to a person, but it is actually acquired from somewhere else. Now, there's another key word that comes in as we're looking at this one, and this is the word pathogen. So as you're going through writing notes on this one, try and make sure that you're putting down the definitions as well. You can always go back and watch this video again later on, or you can supplement it using sort of revision guides and other resources as well. I'm not going to go through and write them all in because I think it's really important to try and do it in your own words and get your head around it, and I'm more than happy for you to come and ask me questions afterwards and go, is this right? I've written this down based on what you've said, have I got it right? Because then I can have a look at what your understanding is. So I'm not going to go through and write them all down word for word. So a pathogen is any microorganism that causes disease. Okay, so we are surrounded and we are covered in microorganisms all of the time. I read one statistic a little while ago that you have more microorganisms in and on your body than you do actual body cells making you up. So if you think about all of your body made of body cells, you have more bacteria on and in you. And that's because bacteria are actually a lot smaller than us, so we can actually fit lots on. And actually we rely on it as well. Morning, Michael. We rely on it as well. So our digestive system relies on having certain amounts of microorganisms in our gut to help us digest our food. And if we don't have that, so if we have some particularly strong antibiotics, then what we actually find out sometimes is that we struggle to digest food. We end up with a bit of an upset stomach until our body can repropagate, until we can regrow those microorganisms. So microorganisms aren't all bad, but the ones that are bad, the ones that cause disease, are caused pathogens. So that's one of those keywords we need to know. So for the rest of this video, I'm going to try and use the word pathogen where I can to try and indicate that we are talking about diseases that are caused by microorganisms. Morning, Chris. So if we just move that up a little bit. Now, there are four main types of pathogens, microorganisms as well, but four main types of pathogens that you need to be able to talk about. You need to be able to talk about some of their properties, some of their characteristics, and I'm going to give you a couple of example diseases that you need to be able to remember. Now, remember, the exam board can and will ask you questions about diseases that I'm not teaching you now. And that's perfectly normal, because if you know about the properties of different pathogens, morning Olivia, if you know about the properties of different pathogens or and how they cause the disease, you can apply that knowledge to any disease that the exam board might ask you. And I'll give you an example of this in a moment. So the first one we're going to have a look at, and I've already mentioned it, is bacteria. So remember that not all bacteria are bad. We rely on bacteria. You probably have millions, if not trillions, of bacteria on your skin at this moment in time. And that's perfectly normal. Okay, we do learn to live with it. Sometimes they can get inside, they can cause a small infection, they can cause spots, etc. But they're not normally particularly much of an issue. The one that we find on our skin most often is one called Staphylococcus aureus or Staph aureus. And like I said, we learn to live with it. If it does cause an infection though, it is usually easily treated. And I'm going to talk about what happens when it's not easily treated a bit later on. So we're looking at now at bacteria that's going to cause diseases. So bacteria are smaller than cells. So we're looking at smaller than animal cells, smaller than plant cells. And you would have actually have covered the structure of bacteria when you did Key Stage 3. So we'll just draw it out again. And I think, in fact, we probably covered it in Biology 1 as well. So feel free to go back and have a look at one of those videos. But we're looking at a sort of basic cell. It doesn't have to have any particular shape. We actually end up with a cell wall. We have a cell membrane. They may have a flagella, which can help them move, a bit like the tail on a sperm. So they may or they may not. They will often have genetic information. But notice it's not a chromosome, it's not in a nucleus. And they will also have other genetic information in little circles that we call plasmids. Now, we know these are smaller than normal animal cells because they're too small for a lot of cell organelles. In other words, we cannot fit mitochondria into bacterial cells. So you won't find mitochondria. So respiration will happen in other ways. So it still happens in bacteria because they still need to release energy. They're still able to make proteins because one of the ways they make us ill is to release particular toxins, which is a protein that they have made. But they are too small for mitochondria. So it gives you an idea of the sort of roughly the sort of size that we're looking at here. Now, what you will have as well is on the surface of almost all pathogens, we end up with sort of protein type structures of some form, and these will be called antigens. And I'm going to talk about antigens a bit more when we get to the immune system. But they'll be sort of surrounding it, and they're unique to the pathogen that we're looking at. So we're looking at the fact they're much smaller. Now they need a cell wall because actually they have no other form of structure, and the cell wall helps stop them bursting. So if they go into a solution which is hypertonic, so if we're looking at a solution, we have a high concentration of water around the outside, hypotonic, with a high concentration of water around the outside, water's going to move in via osmosis. And if that happens, if it didn't have a cell wall, the bacteria would burst. So we know that doesn't happen, so they do have a cell wall. And obviously they need to have a cell membrane because they need to control the substances that's moving in and out of the bacterial cell. But they don't have a nucleus. Now, one of the other key things about bacteria is they reproduce about every 20 minutes. So this is in ideal sort of conditions. So assuming they actually have enough food, they have enough nutrients to provide them, so they can have enough nutrients to provide them with, say, amino acids or protein structures, with enough energy for respiration, with the things that we normally think about, then they're going to reproduce every 20 minutes. This would normally be under an ideal temperature as well. And luckily, if they get inside the human body, luckily for the bacteria, not for us, the human body is at 37 degrees, which is the ideal temperature for respiration. It's the ideal temperature for reproduction. So if we think about this idea of reproducing every 20 minutes, we don't need very many to start with in order to end up with a huge number very, very fast. So I'm going to give you an example of this. Let's just say, for instance, that we started off with one bacteria. So we're going to have one. I'm going to have a look at, and those of you that I've taught this to before, you'll have seen this before, but we can start having a look at just how quickly they reproduce. Now, bearing in mind how small these are, it's highly unlikely you'll ever have just one bacteria. You're probably going to have a few thousand to start with. But let's just say we started off with one, and we're going to multiply that by 2. So in 20 minutes, we'd have 2. That doesn't sound too bad. Now, if we then go to an hour, we'd only have 8. 2 hours, 3 hours, 4 hours, 5, 6, 7, 8, 9, 10, 11, 12. So in half a day, our one bacteria would be looking at reproducing, and we would end up with 68 billion bacterial cells. So we've got this really, really rapid reproduction, and that's just in 12 hours. And obviously it does rely on ideal conditions. It doesn't take into account any of the bacterial cells dying through either old age, through being attacked, or anything else happening. This is just ideal circumstances if we just looked at the growth rates. Now, just for the sake of doing it, we can have a look at 24 hours on this one. So that was 12 hours. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So we're now at a number with 21 zeros on the end, 4.7 times 10 to the 21 if we're looking at standard form, and that would be in one single day. So the reproduction rate of bacteria is huge. Just get rid of that. Brilliant. Now, luckily, our body is quite well designed to fight it and to try and prevent it, and we are going to have a look at that as we go through this particular topic, but it is something that you need to be aware of. Now, the two examples that you need to be aware of that the exam board actually specifically mention is Salmonella and Gonorrhea. So we're going to go through and have a look at the symptoms of those, and we're going to talk briefly about the treatment of those, but bearing in mind any other bacterial infection they can also talk about. So if we start off with Salmonella. Now, it's not as common anymore, so most poultry, which is the most likely cause of Salmonella, is actually vaccinated while the chickens are growing up. So while the chickens are growing, to try and reduce Salmonella getting into the food chain. It's easily killed off by cooking food properly. So if we properly cook food, then it's killed off very, very quickly, and it's at very, very low risk. But if the Salmonella does get into our body, and it gets into our gut, it starts multiplying very, very quickly. And like most of the other sort of bacterial pathogens, it produces toxins. So it's not the bacteria itself which is generally making us feel quite rough. It's the toxins they produce. So these toxins are going to produce a fever. In other words, they're going to raise our body temperature. And as you'll see when we get onto the homeostasis topic on, I think it's biology 5, our body cannot function at high temperatures. It doesn't like it. The enzymic reactions don't work properly, and we start feeling properly rough. And we actually only need to go 1 or 2 degrees above 37 before we're feeling really, really quite poor indeed. So they're going to create a fever. It's going to create vomiting. It's going to cause diarrhea. And we're also going to be looking at things like stomach cramps. Now, during that time, you are going to feel really poorly. But luckily, it doesn't last very long. So normally within 7 days, it's all gone. It's completely gone. Okay, but it would be contagious. So it's from food. It's from not cooking the food properly. It could be caused by if you had raw chicken on a kitchen worktop, you didn't wipe the worktop down properly. I've personally had it but I actually got it from rice. Rice is probably one of the worst foods if you do food hygiene qualifications, which, by the way, you can do online. It's well worth doing. If you do them, you'll find that rice is one of the worst foods that you can actually have for food poisoning, because it's very, very small. It has a very, very large surface area. It's generally quite moist. It's got a high sugar content because it's quite starchy. So it's an ideal breeding ground for most pathogens. So when it's cooked, it's absolutely no problems at all. But when we're looking at savoury rice and reheating rice, we've really got to make sure that actually it's been stored properly in the fridge and it's cooked properly again afterwards if we're going to do it that way. Because it is an ideal breeding ground for most bacterial pathogens. So that's Salmonella. Now, the other one that the exam board seemed to have liked asking questions about, and I'm sure it's just because it freaks out students somewhat, particularly when you start asking about the symptoms for this one, is gonorrhea. Just checking to make sure I've got the spelling correct. So we know that gonorrhea is a sexually transmitted disease. So it gets counted as an STD or an STI, depending on how you want to have a look at it. Sexually transmitted disease or sexually transmitted infection. Which means it's easily prevented by some form of barrier, such as a condom. So from that point of view, it is very easily preventable. However, once it gets in and it gets infected, again, it's going to release toxins. When you look at the symptoms for this one then, we are looking at pain when urinating. And the best one that they wanted in the exams was you end up with a thick, greenish discharge from the genitals. So not the most pleasant. So those are the two symptoms that the exam board will expect you to know, okay? Pain when urinating, thick green discharge from the genitals. Now, it's easily treatable with antibiotics. So we'll put that down. And the one of choice, I'll put that down there, okay. The one of choice, assuming that you are not allergic to it, is going to be penicillin. It's worth bearing in mind with this one, and we will talk about this a bit more when we start looking at drug development later on. And again, when we look at one of the evolution topics. But in the same way that we've ended up with MRSA, there is also some super bug forms of gonorrhea. In other words, because bacteria reproduce so frequently, there is a high chance of a mutation happening. Now, if one of those mutations, and it has happened with gonorrhea, makes it resistant to penicillin, we then need to find other forms of treatment for it. So the more that gonorrhea gets passed from person to person, the more it's allowed to breed and reproduce, the more likely it is that we're going to end up with a resistant strain. So one of the concerns would be that actually we've got a strain now that's resistant to penicillin. If that mutated again and became resistant to another form of antibiotics, we could get to the stage where we actually end up with an untreatable form of gonorrhea. So at that point, you would just have to hope that your body would fight it off itself. Otherwise, things are going to get very, very unpleasant for you quite quickly. So the easiest one, in this case, to make sure that actually we are not passing it on, we're not spreading it, would be this, when we're looking at some form of barrier for it. So I am just going to highlight that, because that's the sort of one that the exam board's going to be expecting you to be aware of. Second after that, though, would be treatment with antibiotics. Okay, so those are the two main ones for bacteria. Again, make sure you know them, make sure you know the symptoms, make sure you know the structure of bacteria, and most importantly, how is that structure different to normal animal and plant cells? So we're looking at the size difference, we're looking at the structure difference, because actually on a foundation paper, one of the simplest things they could ask you to do is to compare a bacterial cell with a plant, bacterial cell with an animal cell, and realistically on a foundation or a higher paper, they could ask you to do a 6-mark question getting you to compare the structures of those, in which case you could literally just do bullet points of similarities and differences. So if you know it, that'd be a very, very quick one to write out. We wouldn't need paragraphs. We wouldn't need massive essays on that one. We could just go and literally bang out the differences and the similarities. So the next one we're looking at is viruses. Let me just bring that size back down to something actually a little bit more reasonable. There we go. So we're looking at viruses. Now, I'll just drop in an image quickly here, showing the structure of it. The image is quite big, and I'll talk about the difference in size in a moment for this. So let's just drop this one. This is actually taken straight from the AQA exam board, one of their past papers. Saves me having to draw it out. Notice we still have some genetic information. Okay. We've got these proteins around the outside for binding. We've got a protein coat. We've got these, so you've got, notice these spikes containing enzymes to break down the host cell membrane, and I'm going to talk about why that's important in a moment. They will also have antigens on the outside as well. In other words, there will be proteins that get recognised, hopefully, by our immune system. So this is the general structure. Now, the really big thing here is they are much smaller than bacteria. Now, it will be easy for you guys to go through, and let me just bring this up on your screen so you can see very quickly. But when you're going through and having a look at these, just make sure that you can do your own research. You can go through and have a look. I did a very quick Google earlier. And let me just see if I can bring this up. Yep, there we go. If you do a quick Google on the size difference between pathogens, or the size difference between cells, bacteria, etc., we can see here. Plant cells, we're generally looking at 100 micrometers. Animal cells, we're looking down at 10 micrometers. Bacterial cells are 1, so it's 10 times smaller than our animal cells. But when we look at viruses, we're looking between this 10 and 100 nanometers. And if you remember, when we've looked at this in some of the chemistry topics, and I'll put it down again here because it's quite important. If we have 1 meter, that's going to equal 1,000 millimeters, which is going to equal 1,000,000 micrometers. So 1 times 10 to the minus 6. Make sure, I'm going to put that over here because you need to make sure that you can do standard form, which is going to equal 1 billion nanometers. Okay, so 1 times 10 to the minus 9. So as you're going through and you're looking at these different sizes, we're looking at a virus being somewhere, if I put those I just realised you can't see that because I did it on the whiteboard. I'll show you that in a moment. The virus here is between 10 and 100 nanometers. Bacteria is 1 micrometer. Okay, so we're looking at the viruses being over 100 times smaller than the bacterial cells. So just to show you what I wrote down a moment ago, if you look over here, and these will be on the notes that I hand out, we've got, you can see the different size difference, say 1,000 millimeters in a meter, million micrometers in a meter, and 1 billion nanometers in a meter. And make sure you can do the standard form on that as well. So the other thing that we know about viruses, which proves they're so much smaller, is they actually reproduce inside cells. So you notice down when we're writing down here, and I'm just going to change the colour just so I can highlight some of the key bits of information on here. So we've got enzymes to break down host membranes. And what they do is they actually inject their genetic information inside our cells, and they allow our cells' normal ribosomes and normal structures to reproduce those viruses. So the viruses are actually recreated inside the cells. And when they actually multiply, so those numbers are big enough, they cause those cells to burst open, releasing more of the viruses into the system. And they can then go and infect other cells. And it's that causing the cells to burst open that makes us feel so rough, because we end up with a lot of damage to cells. And if you think about when we have a normal cold, we produce a lot of mucus, we produce a lot of other, we're sort of constantly coughing, and a lot of this is going to be down to waste cells that are being produced. And again, we're going to get fevers, we're going to get lots of other symptoms that go with it, depending on what that virus is. So we'll just bring that back down there. So there's 3 main ones we need to know for this. One of them's in plants, and I am going to be doing a whole session on plant immune systems later on. Not in this session, but I will talk about what the example is that you need to know. So the first one is going to be measles. Now, generally speaking, measles isn't something that infects us very often anymore. It is dangerous. It's caused a lot of fatalities. It's caused a lot of disfiguration of children in the past. Now, we will talk about vaccinations, but it's easily treated with the MMR vaccination. Now, vaccinations for some people is quite controversial. And unfortunately, a lot of the media, a lot of the newspapers are responsible for this one after a doctor produced quite a lot of fake science on this one, a lot of fake research. The doctor was called Andrew Wakefield, and there's loads of documentaries, there's loads of information on the internet, and I thoroughly recommend actually reading up on it. Unfortunately, and this is one of the reasons why I think people need to learn science, because you need to have enough knowledge to be able to look at information that's given to you in the news and work out how much of it is the truth. But the newspapers, in order to get a story, pushed out the fact that this MMR vaccine, there was a possible link between it and autism. Now, there's been studies since, so if you look at I think it's the Wakefield study in Denmark, where they looked at thousands and tens of thousands of students who categorically produced enough evidence to say there is no link, but that didn't get reported in the newspapers. So lots of people didn't vaccinate their children because there was these concerns between that and autism. And actually, you've had areas now, particularly in some of the smaller villages in some part of the country, where you had quite a large number of people not vaccinate their children. You have had children that have ended up with measles again, and some of these children have died. They've lost limbs. They've ended up with some brain damage as well. So worst case scenarios on this one, but that would have been avoided if the parents knew a little bit more science, didn't believe everything they read, and to make a better informed decision to this, and had actually vaccinated their children. But we'll talk a bit more about vaccinations later on, and we can go through, and if anyone's got any sort of issues with any of this, please feel free to sort of type it in, or ask me some questions later on. But vaccinations from a science point of view are really, really important. So it's a viral disease. It gets in the system. It can cause issues with blood flow. It can cause spots. We can end up with fevers. Have a look online very quickly, and you'll see there's a lot of pictures you can find that give you information on what the disease is. So once we've got it, there's not a lot we can do apart from treat the symptoms, because actually we do have some antiviral medications, but because viruses live inside cells, it's very, very difficult to treat them without damaging the cells that have been actually infected, because the medication can't get in. So we find it very, very hard. So MMR stands for measles, mumps and rubella, Tanishka. So it's 3 separate vaccinations in one, which we get vaccinated against. Like I said, I can go through more about that later on, but it's well worth doing the research. Have a look at the evidence in particular that the newspapers were reporting on, because when you look at the original Wakefield study, a lot of the evidence was just based on 12 children who already had autism, and the link they found was they'd all had the MMR jab. But when you start looking at cause and effect, then to be fair, they'd probably all had breakfast, they'd probably all had a birthday cake at some point, they'd probably all had a walk in the sun at some point. There was no categoric link that it was the MMR jab, but it was something he found in common. So that was literally the extent of the evidence that was put forwards, but unfortunately the newspapers being what they do, and you've only got to have a look at a lot of the news that's being reported at the moment, they'll report anything to get a news, and whilst they don't always categorically lie, they put enough of a spin on it that they can make it sound convincing. So it's really important to be able to look at newspaper articles, look at the evidence they give you, and be able to make your own decision. And if you're not sure, the really important thing is where can you go and how do you research it for yourself? So going online, looking at other YouTube videos, looking at fake articles that are trying to dispel fake news, going and finding different websites, and looking up that information and understanding what you're reading. Okay, can't go over enough how important that is, because personally, from that point of view, if you chose not to do it and your child went down with it, how would you feel at the end of that? That is really that's a difficult one. Very, very difficult one. So we've got this idea of measles then. So that is one of the viruses. The other issue the other disease that you need to be able to talk about is HIV. So we know that HIV is a virus which affects the immune system. It suppresses the immune system and makes it much more likely for us to be susceptible to other pathogens. Now, it's preventable. It's usually transmitted by an STI, by sharing of bodily fluids. Some people have related it back to using needles. There's been all sorts around it, but it's not that uncommon. It's not yet treatable, although there are more and more news articles coming out with science that people have had some success. Now, I've not researched into these enough to find out whether they're reproducible and quite what's involved. But there is some really interesting science at the moment based around this, and it's worth keeping on top of the science and doing the research around it. If you do end up with HIV, however, there is medication now which can suppress it and stop it spreading any further. And many people go on to live a very, very long and normal life with HIV. So the best way is obviously preventing getting into the body. However, after that, there'll be various drugs designed to stop it getting worse. Now, we do know, though, that the worst case scenario is HIV can develop into AIDS. Now, AIDS is not the disease. It is a condition that is caused by the disease, and it stands for Acquired Immune Deficiency Syndrome. So that's the other case in humans that we need to be aware of with this one. The other disease caused by viruses that you also need to be able to talk about is in plants, and it's the tobacco mosaic virus. Okay, and I'll just drop a picture in here that I think I took off Wikipedia actually, which, whilst I don't use Wikipedia very much for research, there's some quite interesting pictures on there. So that one there, we'll just drop it in. If you notice, we get this disfiguration of the leaf. So you've got these sort of pale areas where it's low on chloroplast. So let me just change colour pen so you can see that. So we've got this sort of discoloration. Ooh. We've got the discoloration where there's going to be less chlorophyll and less chloroplast there. So because we've got less chlorophyll, the rate of photosynthesis is going to be less. Now, pretty much with all the plant diseases that you get at GCSE, most of them in some way will affect the coloration of the leaf. And whenever you get discoloration of the leaf, you're going to end up with less photosynthesis, which means you end up with less glucose being produced. Now, because we know that glucose is used for respiration, for producing proteins, for producing fats, cellulose, cell walls, everything else that plants need, if we get less of that happening, then we're going to end up with a plant that's going to be stunted in growth because it just doesn't have the resources to build itself. And we get other effects to the leaf, like it can sometimes curl. But generally, we're looking at a stunted growth. So if you get this in the exam, those are the sort of things you want to put in. Discoloration of the leaf means less chlorophyll, so a lower rate of photosynthesis, so stunted growth of the plants. It doesn't just have to be in tobacco plants. That's where it was quite common, but they can show you this in other plants as well. But you do need to be able to recognise that you get the sort of pattern on the leaves, which is generally tobacco mosaic virus. You'll see the difference in a minute between this one and the other type of plant disease that you also need to know. After that, they can ask you about other diseases, but they'll generally give you enough information to be able to link it either to a viral disease or a fungal disease, based on the information they give you. Okay, so let's go back to black. Those are the first two types of pathogens, so bacteria, and we're looking at viruses. The third one that you need to be able to talk about is a fungal disease. Turning pink will generally be, I think, it's some other deficiency, Tanishka. I'm not a really good one on plant diseases. It's worth having a look though. If you just Google it, now a lot of the time you end up with yellow, and yellow will either be a disease or it'll be a magnesium deficiency, because in the same way that when you're looking at humans, we have iron as the centre of red blood cells. So if we're anemic and we don't have enough iron in our system, our body struggles to produce red blood cells. Plants need magnesium for chlorophyll, so you can often get magnesium deficiencies. In terms of what's causing it to go pink, I'm not entirely sure. It could be a deficiency, it could be a disease. In fact, again, very quickly, we can have a quick look, because it's the sort of thing that's well worth getting into the habit of doing. If I just bring up this page, let's have a quick look at... Let's have a quick look, see what comes up on Google when we have a look at it. Plant deficiencies, why are they turning reddish colour? There you go, nutrient deficiencies. So straight away, we can go through poor soil, maybe insect damage, too much, lots of different issues there. So you can have a look for your plants, but get into the habit of researching these. It doesn't take very long, and we can quite quickly go through and sort of find that out. Generally, though, it's not caused by a pathogen. Right, let's go back to where we were. So, we don't need, in humans, to be able to put down fungal diseases. If you want to know one, feel free, go and have a look at athlete's foot. The pictures are particularly grim. It can affect quite a few athletes who maybe don't look after their feet, but it's easily treatable with sort of fungicides. But the main one that we need to know from the exam board point of view is TMV. Sorry, not TMV. Rose black spot. TMV was tobacco mosaic virus. Now, I'm going to drop another picture in for this. And I'm going to put it directly underneath so you can see the difference. So the TMV is caused by this sort of pale discoloration. When you look at rose black spot, it should be very, very obvious. We've actually got now properly dark discoloration. This isn't a fading of the leaves. This is pretty much where part of the leaf has died, because it looks like the way plants go in the winter, but in very localized areas. So the symptoms are going to be the same. We're going to have less chlorophyll, we're going to have less photosynthesis happening, which means we're going to end up with stunted growth. In the same way that TMV doesn't have to just be on tobacco plants, rose black spot doesn't just have to be on roses, it can be on other plants as well. So they're not going to expect you to be an expert on identifying plants based on leaf shape and plants. It's just the name given to that type of pathogen. With all fungal diseases, they're spread by spores. So spores are like their seeds, but they can generally be, you get lots of them, as in thousands of them can be spread very, very quickly. So in terms of the treatment for these, usually we want to destroy them or use a fungicide, which is a particular chemical that kills off fungal pathogens. But if we haven't got a fungicide, we can remove the infected leaves, and then we can just destroy them, we can burn them, we can get rid of them. We can do the same thing to the leaves that are infected with TMV as well. But the main thing is that you want to try and do this before we've had time for those spores to spread, because once they've actually been released from the plant, they can spread very, very fast indeed. Now, the last type of pathogen then, and in some respects is one of the most important, although it's not the most common to us. We don't see it as often where we are. In other parts of the world it's more prolific. But this is a protist. Now, the reason it's more important in a lot of ways is that the example of a protist that you need to be aware of is malaria. Now, there we go. Malaria is really, really important because the malaria protist itself is responsible for more human deaths than every war in the history of mankind combined. So we are looking at hundreds upon millions of thousands, huge amounts of deaths throughout history. So it's quite a scary statistic when you look at that. But we are looking at this idea that it's responsible for more human deaths than every war in the history of mankind combined. Now, it relies on the mosquito being the vector. So part of the life cycle of the protist, of the malaria protist, is the mosquito. And if you eliminate the mosquito from the life cycle, the malaria protist cannot survive. So mosquito is part of the life cycle. If you get infected with the malaria protist, you have it for life. That doesn't mean you constantly have the symptoms. Symptoms do include fever and other symptoms as well. And it can be fatal. But you actually end up with recurrent bouts throughout your life. And that's because the protist itself can live in spores in the liver. And then periodically through your life, what will happen is those spores will be re-released back into the system and you'll end up being infected again. You'll have to be treated. And it can be very, very dangerous and potentially fatal. So again, the best way of dealing with malaria is prevention. And I've seen quite a few exam questions on this in the past, asking people to talk about how you would prevent it. So the first way is avoid being bitten. So we could be looking at bug nets. And that could be around us during the day. That could be around the beds that we're sleeping in at night. We could be looking at chemicals. So by that, we're looking at things like DEET. Or if we're looking at our clothing, we're looking at other chemicals like permethrin. You don't need to know the names of these two. Permethrin is quite a good one for treating clothing, depending on where you're going. DEET is one of those chemicals that I don't particularly like using, but it is so effective at stop getting bitten. And there aren't many other things out there. There's lots of natural remedies. There's lots of alternatives that people have tried, but I haven't personally found one yet that's as effective. And I spend quite a lot of time outside, and I'm quite prone to getting bitten by all sorts of things. So we can use chemicals on that side of things as well. They have in the past tried widespread spraying to kill off mosquitoes. Unfortunately, the chemicals they used at the time, and I'm going to talk about this in a later unit when we look at ecology, but if you want to do some research, have a look at DDT. DDT was a chemical that they sprayed back in, I think it's the 40s or 50s, I can't remember the exact time span, and they did massive widespread spraying of certain areas with this chemical that was very, very effective against mosquitoes. Unfortunately, DDT is a fat-soluble chemical, and it ended up in the food chain, and it's remained in the food chain for many, many, many years. And what it did do is it started causing other issues in the food chain because it bioaccumulates, and it caused things like bird shells becoming weak. So when birds of prey particularly sat on the nests, the shells are breaking. They found it in sea life. They found it all over the. They found it everywhere. So same person being affected. Once you're infected, I'm pretty sure you've just got it, Tanishka. I'm not entirely sure, but I'd have to double check. I don't think it gets any worse if you're multiply infected because you've already got it. It might just mean you have another bout of the infection there and then. Interestingly, and this is something that when you look at it, there is some evidence that if you look in some parts of the world, and I'm just trying to think, some people have sickle cell anemia. Now, this hasn't been in the spec for a while, but it wouldn't surprise me if it came up. So I'm just going to talk about it briefly now. Sickle cell anemia is not a communicable disease. It is a genetic disorder, and I'll probably talk about this again when we look at genetics. And rather than the red blood cells being sort of, if we look at them sideways on, being this shaped or sort of rounded, when you look at them, they generally form slightly odd shapes. which means that actually they can get caught up in blood cells. It can be quite painful to the people that have this if they start doing strong exercise. But because the blood cells are weaker, they can't support the malaria protist. So I'll do a bit more digging around on that one to find out whether you still need to know about it. But actually, it's one of those things that in some parts of the world, the sickle cell anemia gene has become more common because it's given people immunity to the malaria virus. So it's almost a form of evolution for those people. The white chemical released in the air helps in killing mosquitoes. It could just be some form of insecticide. I don't think they've got one yet that's absolutely specific to mosquitoes. Now, one of the things they are looking at doing, and there's lots and lots of research that's being done on this, they've actually looking at doing some genetic modification, genetic engineering of mosquitoes. And I will talk about this again when we look at genetic engineering, because I know some of you want the debate on this one, and hopefully by then we'll get a few more people being online enough time to actually do this. But what they're looking at doing is genetically modifying some of the mosquitoes to be sterile, then releasing them into the environment, because whilst they can't breed, I think it was generally the male mosquitoes they were looking at modifying, but by releasing them into the wild, the females are going to be still breeding with those ones, which means we're not then producing live mosquitoes, so the numbers of mosquitoes are going to be dropped. So that's one other thing they're looking at doing. Another form of treatment or another form of prevention is when we're looking at medication. So for us, if we go to parts of the... I know you can't. I tell you what, if you can get enough of the rest of our class and some of the other classes to regularly start coming onto Google Meet, I will organise a debate. But at the moment, there's not enough of you to warrant it. So I'll leave that challenge with you. But hopefully, with some of the other stuff that's coming up at the moment at school, we should be able to get a few more people online. But I think that'd be quite good fun. Yeah, so medication. So we can take anti-malarial medication, and what we normally have to do with that is take it for a certain amount of time before we go to areas where malaria is prolific. And then we'll take it for a certain amount of time when we get back, and we carry on taking it while we're out there, which means while we're in those countries, we cannot get malaria because that drug avoids us. It avoids, if the malaria does get into our system, it's killed off straight away, and it doesn't manage to get a foothold in the body. The problem happens is that some people stop taking the medication when they come home, which means the protist may have got into their system, and they stop taking the medication, which means it can then contaminate or infect that person, which means the person still has it. So if we go to countries where we know that actually malaria is fairly prevalent over there, then we need to make sure that we actually have the medication and we follow the safety advice to avoid actually getting the disease. Okay? So those are the main types of pathogens that you need to know about. If you want me to go through any of those again, that's absolutely fine. And I'm more than happy to go back through and we can talk about it in Google Meet. You can ask me questions online and we'll go through it again. What I'm just going to finish with, because we've got about... In fact, no, I'm not. I'm going to do that next time around. I think I'm going to finish the video there because we've got 50 minutes. So for the remainder of the 10 minutes of the lesson, I'm going to go on to Google Meet and I'll answer any questions on this one. And that means on Wednesday, when I do the next part of the video, we can then start looking at the ways our body prevents being infected and what happens to the body once we have become infected. Okay? So we'll call it 50 minutes. And in the next 2 to 3 minutes, I will post a link on Google Meet and we can have a chat and we can meet up and go through any of that. So those of you online, keep an eye on Google Meet. I'll be on there in the next couple of minutes. Good to see you all online. And thanks for the questions. Take care.